Good morning, my name is Kristen Ellingwood and here's my project. And it's about receptor tyrosine kinases. So receptor tyrosine kinases are receptor proteins in the plasma membrane that start out as inactive monomers. Okay, so here's our little cellular membrane or plasma membrane, and this is our monomers, and there are two separate monomers that are sticking out into the cytoplasm. This is our ligand molecule or signal molecule, which fits only into a specific receptor protein. And in this instance, it will be the tyrosine kinases one, which is currently separated right now in two separate monomers. A kinase is an enzyme that catalyzes the transfer of phosphate groups, which will be later seen in stage 3. So for now, we're going to continue on into stage 2. So there are two signal molecules that are needed for the monomers to come together, as you can see here. They have now changed shape, which allows for them to be activated and eventually sell a signal going out calling for phosphates. These typically are used to regulate growth factors in organisms. They can stimulate molecules to grow and repair tissues, and in some instances even cause organisms to increase in size. So now that the dimer has been created, this is a dimer which is two monomers that came together, so you can kind of hear it in the name. And so now that the dimer has been created, it is activated, it can be phosphorylated, which means to add a phosphate to a protein such as ATP. And you can see that 6 ATP were put here, and then they were changed for the reaction. And so now that the dimer is completely activated, they can interact with other molecules that are out in the cytoplasm that we are currently unable to see. So now that we have the fully phosphorylated receptor, it can be recognized by multiple proteins. These are two different these are proteins that are called relay proteins, which are part of transduction pathways, which were introduced earlier into the textbook if you read it. These receptors can trigger separate cellular responses. For instance, this protein can go off and have it do one response, and this protein can have it go off into the other side of the cell and have it doing a completely separate interaction with the protein, or sending a completely separate message that has nothing to do with this one. So now, and the cytoplasm is completely full of these relay proteins, and so all of them race there to get the information that is needed, so they can race back out into the cell and conduct whatever needs to be done. And these proteins are always waiting to be ready to be switched on. It's kind of like right before a race, you're ready just to get out there and get it done. Oh, random fun fact, many cancers are actually caused by mutated receptors that are activated without a signal molecule, which is just completely fascinating by my standards. So the receptors that we had seen last time could have been used to help maintain homeostasis. Homeostasis is the ability of the cell to keep it internally stable. So, and there's several different stages for a cell to maintain homeostasis. So there will be stimulus in the so there would be stimulus inside the cell which would change a variable, and you would be able to see that the cell inside the cell there would be receptors which would be able to detect the change, and then they would send information along a completely different pathway to help fix the problem to the control center, which is then sent along a different pathway to the effector, so that way you can send the information along even more. And so then the response of the effector feeds back to influence the magnitude of the stimulus, which returns it to homeostasis. So in other words, it just makes it equilibrium again. And then the cycle just repeats back and forth continuously. It's kind of like the circle of life. And there are different, so cell signaling isn't the only form of homeostasis in a cell. There are other instances where homeostasis does not need cell signaling. For example, temperature regulation is controlled by the brainstem, and you sweat and shiver to help maintain a certain body temperature. And then blood pressure allows for osmosis and diffusion between cells, and that is just completely all on its own. That has no influence between the cell whatsoever. And fluid balance, again, on diffusion and osmosis. So there you have it. So you'll also later be asked about hormones in a cell. And hormones are used for long distance signaling, and the hormone is released into the bloodstream so that way it can reach its target cells, and they will collect the information and then respond to the hormones in whatever manner it is, whether it means that you're starting to become a woman, a man, or you're just going to grow even taller. Thank you for listening to my not so good project of this. Thanks for taking the time to listen to this. Sorry for rambling. Bye.